So our myofibrils, they're made up of a bunch of different parts, and we have our um, dark A band, and that's a little overlapping there. So it's a darker portion there, and the lighter I band portion, okay? So our A band, and by the way, I'm trying to figure out where we got these names from. So I haven't figured it out yet, but I will find out for you. But you'll see that within this myofibril, all of these different parts of it has a letter that's going to distinguish a different part of it. So one myofibril is being pulled out here is made up of several different sarcomeres. So here is a sarcomere, and it goes from Z disc to Z disc, okay? Within this, we have I and A bands. So I just wanted to point that out before we talk about A and I bands. So these A and I bands, they kind of create that striation look. And an A band is representing our thick filaments, which I'm going to give you a better look at what this thick filament is in a moment. But look at this red thick line here from here to here. That's all of our A band. So that means all of that thick filament is in this dark A band here, 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 and here. Now the lighter portions is made up of the thin filaments, although our thin filaments do overlap over our A band. So we're going to call that area something different in just a moment. I'll show you. So A band, where the thick filaments are, I band, where only the thin filaments are. That's your take-home message right now. And like I said, a sarcomere, that's the functional unit of muscle, will go from Z disc to Z disc. Okay, so one myofibril is made up of a bunch of sarcomeres. Okay, so let's talk about our thick filaments. Our thick filaments are actually made up of myosin, well, it's a protein, but they're called myosin myofilaments, and they extend through that entire A band, okay? And they're going to have these little heads that stick out. These are our myosin heads. We got that blob, okay, all over here. These are all myosin heads. Those are going to act with, oh, there's our A band, with our thin filaments, which are made up of another protein called an actin filament. Or the actin filament has a protein within it, but this is a myofilament here, represented by the blue lines here as well. And then we have our Z discs. No, that's not what I'm representing for you. I'm representing in this purple box the I bands, where we have only our thin filaments showing, and it stops at the thick filament, which would make up our A band. There's our Z discs. Our Z discs are shown in green, and that is kind of holding on to our thin filaments here, okay? Our thin filaments will move in toward one another when a muscle contracts. So we'll take a look at that in a moment too. We have our M line, and our M line is representing for you, well, actually, now that I look at it, this is representing your H zone. Sorry, I don't have the zone labeled for you, but it's up here if you can see it. And this is when our thin filaments end and our thick filaments are overlapping, okay? And this H zone disappears when muscle contracts. So we have this lighter region here in the middle of our A band. Okay, so we got our thick myofilaments here and no thin filaments. Now our M line, there we go. Now we can really see our M line really well. Our M line is a line of a protein known as myomycium. I'm guessing that's why they called it M line, because of the myomycium, starts with an M, and this bisects our H zone vertically. So it just splits that H zone in half. Okay, next we got more pictures. There's our M line again. I wanted to show you, here's our thick filament made up of our myosin heads, and these are all going to attach to our actin filaments or actin molecules here in just a moment when we're trying to contract. I put this picture here because I wanted to show you a cross section of of our sarcomere. So we can see the arrangement of our thick filaments surrounded by thin filaments. Each one of these is a little bit different. So our I bands, we know that that is representing only our thin filaments. So that's the only filament we see here. Whereas our H zone, we know our thin filaments aren't overlapping there. Here was our H zone in this area. So we only see our thick filaments there. Now our M line, we see our thick filaments linked by accessory proteins, and that's what these vertical lines are representing for us. And then we have the outer edge of our A band, which would be right in this area, where we would see an overlap of our thick filaments and our thin filaments. So you see that down in here. Okay, just a quick little look at our myosin heads. These are actually made up of a bunch of tails and heads. So here is our tail of our myosin, and these would be our myosin heads, and they have these binding sites that are going to bind to actin, and I'll show you what that looks like in a moment. Let me show you this first. So here is our actin, or our thin filament molecule. These blueberry looking like substances, that is our actin subunits, and they each have active sites for the myosin heads to attach to them. Now we have also tropomyosin that's blocking all of these active sites. Do you see how it's covering those holes? Well, they're not really holes, but depressions, I should say, for those myosin heads. And then we have tropomyosin here. Now, when we have calcium binding to tropomyosin, it's going to help shift the tropomyosin strands so it reveals these active sites, and the myosin can then bind to the active sites. Okay? And these are also called G-actin molecules, these blueberry looking like structures here. Okay, so let's get back to our SR and T tubules. Okay, just to refresh your memory, here's the cisternae. We have our tubules sitting in between here vertically, and then I have a couple pictures representing this so you can see it in multiple different ways. So what happens is we get a neuromuscular junction here. We get the acetylcholine neurotransmitters going over, activating a nerve impulse, telling the muscle to contract. That information is going to travel here along the T tubule, tell the SR to release calcium, and that calcium binds to, remember troponin on our thin filaments, it binds to troponin. That's going to shift this tropomyosin strand out of the way, allowing the active sites to be attached to the myosin cross bridge. And this is just another view of those structures here. Now this is going to go a little specific. I wanted to get into this more in this prep screencast, but I want to make this short and sweet, so we're going to skip over this. We'll talk about this in class. Okay, what I want to talk about next is the sliding filament model. So I kind of mentioned this a little bit before, but what's going to happen within our sarcomere, okay? We are going to get a nerve impulse telling our muscle to contract. So when that happens, first our myosin cross bridge that has already attached here is going to release its ADP and phosphate, and that causes this myosin head to do a working stroke. So it's going to bend the head and pull on the actin filament, that's the blueberry-like structure, and slide it toward the M line. So the M line would be right here. Next, what happens is a new ATP is going to come and attach to the myosin head in order to detach the myosin head from the actin filament. After that happens, we get ATP hydrolysis, breaking off one of those phosphates from our, what was before the ATP, and now we have ADP. And so we get the myosin head cocking back and gets ready to attach once again. So when this sliding filament happens, it brings these actin filaments more in. Do you see how they're further apart here? Our H zone is bigger here. When our muscle contracts, that H zone disappears, like I mentioned before. So those actin filaments are being pulled in. So the myosin heads kind of pull onto it, in, pull in, like that.